this talk. Okay, so that if somebody wants to hear it tomorrow or next week's, I will send the link. Actually, I recorded all but one seminar, so there will be. I will send the link so that everybody can listen to them in the next days. So Fabrizio received this in Perugia, in Italy, in 1984, and then he got his HDR from Université Louis Pasteur de Strasbourg in 2004. And after a first part of his career at the Laboratory of Neutron Physics, and where he was working Materials and Chemistry department in Rome, he went for a 4-year term in different US laboratories. And in 2006, he became full professor in Lille, at the University of Lille. And in 2010, he created a 2-year master school of biological and medical physics in the Department of Physics in Lille, of which he is a director now. And since 2019, he is director of the physics division at IEMN in CNRS. And during his career, he has visited several laboratories, including COA, Cadarache, MIT, Kavli Institute for Theoretical Physics in Santa Barbara, etc., etc. So thank you very much, Fabrizio. You can start.
Can you hear me? Does it work? Microphone? Nobody hears?
I do.
Yes, you do?
Yes, Professor.
Fine. Okay, great.
It works. It
okay,
works.
so okay, so thanks for the introduction. And since I couldn't bother you with my weekly letter last Sunday, I decided to bother you with a seminar today. That's the idea. So these seminars are kind of intended also for letting other people in the IEMN and outside IEMN as well know our activities. So my first slide is not about the seminar, it's just to tell you what are our key activities in the Namaste, nanomaterials and soft matter team in the physics group or division. And we do essentially work on molecular cellular biophysics. One main area is radiation damage in DNA, in nucleic acids, and repair mechanisms, on which we have a number of collaborations. One INR that we hope it's just waiting, crossing fingers, one ICIT project, and one H2020 collaboration that touches upon life on Mars. And the second area, which started more recently, is related to the first as far as signalization mechanism, but mostly related to cancer development mechanisms, in which as well we have a smaller number of collaborations. And for all these activities, we perform theory simulation with statistical mechanics models. So what is not in this first two slides is today's seminar, which is another interesting, quite large part of the work, which is about DNA again, but a special kind of DNA, artificial DNA, that has impact also potentially on technology, on technological tools. And the seminar of today is this thing about DNA aptamers for detection of cancer cell, viruses, and near living stuff. You will see at the end, I will just touch upon some more open, larger subjects. So I'm working on this thing with people here in Lille, Ralph and Mark Lenzing and Chan Lagadek in biology department, and people in Tokyo, Nicolas Clément, Xiu Li, and other people. If you want more information on this activity, you can go to that beautiful website of mine and you will have everything you need. Okay, so that's the outline of today. Of course, some introduction. What is an aptamer? How do we know about their structure, stability, and thermal stability, thermodynamic properties? Then I will go into a couple of examples of what we are doing. One example about targeting cancer cells with these devices, and another example about recent antiviral ideas against SARS-CoV-2. And at the end, the little opening about how aptamers enter in the discussion about origins of life, astrobiology, and we will close the loop back to life on Mars again. So to start, what is an aptamer? So an aptamer basically is a small sequence, 10, 50 kilobases, not kilobases, long of a nucleic acid, RNA or DNA, that has outstanding, extremely selective protein interaction capabilities. Parenthesis, one can make aptamers also with peptides, but this is not the subject of today's talk. Target recognition involves the 3D shape of the aptamer. So one starts, as you see on the right of the slide, from a sequence, just a sequence of DNA. This sequence can interact inside the sequence by forming pairs. And then you see here the 2D structure of a sequence, where some bases can pair, some other bases do not, because they are not homologous. So what you end up is with a structure made of hairpins. What you see, these little round things that are like little bones are called hairpins. And this is just the 2D structure, which then, because of steric interaction, hydrophobic, hydrogen bonding, base tagging, falls into a 3D structure. And this is the object that has a very high selectivity for protein recognition, because it's a sort of small mold that is able to recognize, to fit onto some target, molecular target, not typically protein target. RNA and DNA aptamers are pretty much the same. There's a very small difference between the two, but DNA, as in real life, is much more stable chemically. So although the whole thing started with RNA aptamers, today people work quite a lot with the DNA aptamers as well. So the procedure to make, to build aptamers is well developed today. It's a chemical solid state synthesis. Of course, biocompatible, it's just DNA. Label free for a practical application. This is a great advantage. And then very selective. We will see this later on in the talk. Cost effective. DNA, especially DNA aptamers, are suitable for mass production and very cheap to produce. Quite stable mechanically and thermally, so they can be transported easily, they can be packaged easily, relatively easily. And so as far as they go, they can play, well, it's a little bit optimistic, could play a role in detection and treatment strategies for a number of diseases, okay? So you see again here the three-step activation. So start from a simple sequence, go to a 3D structure that then has some specific recognition property and can interact and bind to a biomarker and then do something, either elicit some response or block some response or favor some other kind of interaction, number of things, okay? Sensitivity and selectivity, how sensitive and how selective they are. Well-known diagnostic assay is theophylline versus caffeine. These two molecules that you see in the middle of the slide are practically identical. They differ only in the upper right of the molecule, where there is an NH bond or a methyl CH3 bond that differentiates between theophylline and caffeine. So it's a very small difference from a molecular point of view. And an aptamer that has been created around 25 years ago already, this sequence that you see here, has a very high level of sensitivity, meaning the minimum amount of the molecule that has to be present in order to be detected. And we are less than micromolar. Actually, aptamers can arrive quite easily to nanomolar sensitivities. And the selectivity, meaning that how much you can discriminate between similar targets. You see, it's like 10 to the 4, 10 to the 5 selectivity, recognizing one molecule with respect to the other. So this is a quite clear example of how selective and how strong can be the interaction of these little objects with a molecule. So a little bit of history. So the first notion of selection in vitro was introduced many, many years ago by Sol Spiegelman in Urbana, where he used a phage display technique, a Q-beta, to evolve a self-replicating molecule. This was the first idea of in vitro selection of molecules. Then, a few years later, Gerald Joyce at Scripps started using a system that he called directed evolution to modify, spontaneously modify the activity of RNA enzymes, what is called the ribozyme. In 1990, again a few years later, two labs independently developed a technique for making automatic selection among millions, billions of sequences. Larry Gold created a new process for selecting RNA ligands, which they called SELEX. That's the name of the game. And more or less at the same time, Jack Szostak at Harvard developed the method of in vitro selection, selecting the ligands, RNA ligands, against various kinds of molecules. And it was his student, Arthur Ellington, that invented the term aptamer for these ligands. And in Arthur Ellington's lab, a few years later, Colin Fox developed a new technique to improve the SELEX, which before took several weeks. Now you can do a SELEX selection of aptamers in about 3 days. I put the face on the slide because I want to know these people. And one important thing that links all these people in the slide is that all of them are scientists that are interested in the origins of life. This is their main area of work. So this whole business about selection, a spontaneous selection of defined sequences over pools of billions of sequences is something that is closely related to the theories about origins of life. Structure, stability. How do we know the structure of an aptamer, given its quite complicated folds, and how do we know whether it's stable or not, whether it will survive or die? SELEX method means systematic evolution of ligands by exponential enrichment. So basically, it's a cyclic process, which is illustrated on the right side of the slide. It begins with the synthesis of a large oligonucleotide library. So you take a DNA printer, basically, something that looks like a printer, and generate millions and millions of random sequences of fixed length. These sequences may have something in common, so one may impose that one part of the sequence remains the same and the rest is random, or they can be really totally random, okay? Then incubate these random sequences with your target, the molecule that you want to recognize. After some time, you remove the unbound sequences and keep only the sequences that have been binding to the target ligand. The removal can be done by different methods, by chromatography, by magnetic beads, or by some other methods. And then you go to PCR and amplify these winning sequences, and then go on with the game, okay? And after some rounds, you get one group of sequences, one or few or more group of sequences that have a high affinity with your target. Then there are other techniques that can be used to refine the procedure, but the basic procedure looks like this, and it's almost completely automatic. So which is the structure that corresponds to a given sequence? Because what you do is just plug in the sequence that you want, and then you get out something that binds to a molecule. How do you know how it works? So for example, let's take a sequence like this that is supposed to recognize another kind of molecule, the protein, the serpin. This is just a sequence, and looking at the sequence, one can see that there are here and there some complementary pieces of DNA that could form some bond, some bonding. And actually, you can use different methods to estimate what could be the 2D structure of this object, so which bases can and will pair once in solution. And there are models to try to understand these, different models, M-fold, NUPAC, and others. They don't always agree because some differences of free energy can be very tiny, very small. So depending on the database, depending on the refinement of your structure, one can get kind of close looking, but not exactly identical sequences. So you see, for example, for this A12 aptamer, I ran 3 different runs, and you get 3 structures that are quite close in free energy. You see the yellow portion is the one that is complementary. The rest is hairpin or open loops. So they don't pair. And curiously, you see that, for example, the structure on the leftmost side has 5 GC pair and 3 AT pairs. The structure in the middle has 7 GC pairs and 3 AT pairs, but has more mismatches. So even though it has more bonds, it has a lower, higher, let's say, free energy. And the one on the right is very similar. It has even less paired bases, see, but is supposed to have an even lower free energy. That's because of base stacking and other hydrophobic interaction that you don't see here, because this is just a 2D structure. You should see this in a 3D structure. When you go to the 3D structure, you may ask, how stable is this structure? Will it just unfold in the matter of a few minutes, or if I just change a little bit the pH of my solution, or if I change the ionicity of the solution and so on? Will it retain the structure, or will misfold while interacting? How long it will last? These are technically challenging questions, okay? Now, the melting temperature, so the temperature at which a DNA or RNA sequence gets broken apart, can be established theoretically on the basis of a nearest neighbor model, which uses experimentally fitted parameters for enthalpy and entropy based on combinations of base pairs and nearest neighbor interactions. So when you look at the stability of the Serpin A12, for example, again, aptamer as a function of concentration of ions, you see that the melting temperature can change by a factor of 50% also, at most, if you use especially divalent cations that have a special interaction with the backbone of DNA. So strong dependence and so increased stability in more ionic solutions. Increased stability because you see the melting temperature goes up. So it means that the molecule the aptamer will be destabilized at a higher temperature. Or one can use tricks, especially with the RNA. One can modify the basis, for example, by making this oxygen-carbon bridge that will increase the rigidity of the coupling of the whole structure, sorry, or protect some bases. Again, the hydroxyl that is present in DNA, in RNA here, in the sugar here, the difference between DNA and RNA is just this, you see, OH and H, but this OH is very reactive and can be protected by replacing it with a fluor or a methyl and so on. Okay, so there are techniques that can improve and increase the stability of these objects. Okay. Now we move to my job, fabricating aptamers in the computer. Okay, so once the SELEX has defined what is the winning sequence and has given a first measure of the affinity, this can be easily in the nanomolar range. One big problem is that, okay, you see that this thing binds to a ligand, is very effective, but how does it bind? Where it goes, where is the interaction? And this you cannot tell from SELEX, you cannot tell from a standard chromatography. You have to go inside the molecule and look what's going on, what's happening, right? So here is where theoretical modeling comes to help. Unfortunately, there is no simple way to predict the 3D structure of nucleic acids or proteins. We're still very far down the road, still in this business, starting just from the sequence. So I give you a sequence and you tell me what is the final 3D structure. So the protocol that we have assembled in our group now is to start from the 2D model and look at competing structure from different softwares that define the 2D base pairing. Then, unfortunately, these softwares work only for the software that give you the 3D structure work only for RNA. So we first convert DNA to RNA, obtain a 3D gas structure, and then go back to DNA. Of course, this is not the ideal structure, but we have no tools to predict the real 3D structure of DNA, so we have to go through this intermediate passage of transforming DNA to RNA and then going back. And then from here, we have a candidate structure on which we can start running molecular dynamics and all the tools of the trade that we use for working with proteins. As a simple exercise, I show you here the comparison of some structures of hairpins predicted by this long procedure, computational procedure, after MD relaxation at normal conditions, so 310 Kelvin and 1 atmosphere. And in red, you have the predicted structure. In blue, you have the experimental structure. So these are 6 small hairpins for which experimental data are available. You see that the structures are quite close, not perfectly close, but there are strong similarities. In some cases, there are some bases that are opening up, but this is normal because this is a very open structure, so bases can fluctuate quite freely, like up here, the blue one, or up here, the red one. In this case, for the 1EN1, the agreement is very bad, so there is almost no agreement. But in the other case, you see that this technique has a quite decent, I would say, level of predictability for the structure when the structure is known. Problem is that this is known for small hairpins, but for complicated aptamers, there is no X-ray or NMR structure available, so one has to just go on and guess, okay? And then when you have a structure that you think is okay, is correct, more or less assessed, then you want it to interact with your ligand and understand where the binding occurs, how stable is the association, what are the molecular effects, what are the modifications that occur upon the interaction, if any, what are the key effects that lead to the stability and selectivity of the aptamer. So in this case, we need to again use a complex protocol that includes going back and forth between 2 different steps. One is the docking step, in which the 2 objects, aptamer and protein, are attached together in many different configurations, and the free energy is estimated by some simplified functional, which often includes also entropy terms. This allows a rapid exploration of the conformation space, and then you get a ranking of the best candidates of the interaction after this space, that then you cycle through real molecular dynamics, in which you can do a virtual experiment with all the bells and whistles, ions, water, explicit solvent, and there are techniques to accelerate rare events like metadynamics and so on and so forth. Then your thing starts to showing some properties, then you can go back to the docking and redock the final structures, imposing constraints, and then repeat the procedure again until you arrive at something that looks quite stable, quite invariant, and so it gives you the possible, let's say, more probable, more likely structure of the interacting couple. Okay, so that's the end of the first part, and then let me move quickly to the 2 examples, aptamers to identify cancer cells in this case. So one typical target is this protein that is called EpCAM, epithelial cell adhesion molecule, which is used as a key molecular marker, both in diagnostic and in prognostics of cancer. So the structure is quite big, quite complicated. The interesting part is mostly the part outside the membrane, which is supposed to make adhesion between pairs of cells. And as you can see, maybe here, it's supposed to be a dimer, also two copies of the same protein coiled together that have this extra cellular part that makes adhesion complexes. So one of the ideas is to block the activity and to recognize the activity of this EpCAM molecule, which is very much expressed on the surface of cancer cells that want to attach everywhere. That's their business. And so a high density of these molecules normally signals cancerous cells. So this is a potential lead for a technique that could recognize quickly and effectively circulating cancer cells in the bloodstream. Now, one aptamer has been identified a few years ago, this F-Cleri, which is a strong ligand against EpCAM. So one can maybe invent some device based on this aptamer that could allow recognizing the cell that has these many EpCAM proteins expressed on the surface. So these are the basic properties of this object. You can see here the 2D structure. There are different hairpins that are stable at different temperatures. So this part here is the less stable. It starts opening up at about 28-30 degrees C. So it should be open in vivo, because in vivo we are at 37 degrees, our body. This second structure is more stable. It opens up at about 42 degrees C. And this one is the strongest one. It opens up only at 50 degrees C. So we can think that this part at least should be indefinitely stable in the human body. Okay, so that's the key message. So in the Tokyo laboratory of Nicolas Clément, there have been invented a number of devices. The one on the left is an older version of the device that they were thinking of. The one on the right is a more recent version of the device. So the basic idea is to have a substrate with an electrode, gold, grown on doped silicon. Then there are some pillars here made in some plastic, basically, on which cells can adhere, repose. And between the pillars, one could place aptamers that could recognize the cells. And what happens is that the current that goes through the aptamer changes when the cell blocks the movement of the DNA aptamer. Now, what you see here is a bad sketch because the cell should be much bigger. So the distance between the pillars is 200 nanometers, you see. So a cell typically is 10 microns. So the cell here is very small, but it should be much, much larger, okay? So the basic idea is to have some detector attached at the end of the aptamer. A detector can be, in this case, a ferrocene moiety that does redox cycling. So going up and down, it contacts with the electrode, and when the cancer cells are above, the aptamer is stuck and it doesn't contact the electrode anymore, so the current changes. Okay, so one can detect current change and think that there is something that has locked in the aptamer, i.e. the cancer cell is there. So the full molecular model that we made is something like this, with 2 electrodes for symmetry, because then we have to apply electric field, so we cannot work with just 1 electrode. In the experiment, there is the counter electrode, which is very far away in the microfluidic cell. Here, of course, in our case, we have to put the 2 electrodes much closer because we cannot simulate the whole microfluidic cell. So then there will be a rescaling because the electric fields that we get here are much higher and so on, okay? But overall, it kind of works. So this is the structure where I removed the water to show you. So here you have the aptamer. This is the most stable hairpin loop, where we attached the molecular species carrying the ferrocene, the green ball, and the aptamer is fixed by a thiol linker at the lower electrode. There are a number, of course, of technical details that I'm sparing you. Basically, what we do, we run simulations to understand how the aptamer moves in this configuration with and without applied electric field. And we try to characterize the frequency by which the ferrocene gets close or far away from the electrode, because then this goes in another modeling program that can tell us how the current should change, if any, when the aptamer is blocked or non-blocked or free to move. So I think, yes, here there is even a small movie that maybe you can enjoy, where you see the water is removed, but it's there. The ions are removed, but they are there. So you see the ferrocene getting close and jumping up. So the vertical excursion is about close to 8 nanometers, which is more or less the height of the pillars in the microfluidic cell. So it means that in this configuration, the aptamer can go touch the EPCAM molecule if the EPCAM molecule were there. And you see the 3D structure, the 2D structure here in green, blue, and red. And here you see reproduced the 2 parts of the structure, green, blue, and red. Okay? So you see that the most stable part of the aptamer is the one that holds the ferrocene. Okay? Okay. Then the next step is simulating the interaction with EPCAM. So we imagine that a cell is up there, some EPCAM molecules are coming from above, and then you had to do the whole binding candidate ligand target conformation again to identify how the aptamer would bind to the EPCAM dimer, and in this configuration, how does the ferrocene ball there move to under different kind of electric fields, and then accumulate statistics about this movement that then are fed into the program that calculates the probability of detecting some level of current. So here I don't have the video, but it should be somewhere here. How do I switch the screen? This place. Mirror. There. So in this case, the video is on my YouTube channel. So one can see. Here we have the aptamer, which is docked to the blue EPCAM protein. So it's permanently stuck. The aptamer has been captured by the protein, and the ferrocene still moves, but it moves in a more constrained way. So we can make a statistics of this movement and compare the statistic of fluctuation with the free aptamer. And this gives us the key information about probability of detecting current, and then getting some signal from this configuration. Okay, so this is basically what is the typical result of our simulations of aptamers in a quite complete, let's say, setting. Of course, the cell is not there. All we are interested is in how the aptamer interacts with the single protein. Okay. Okay. How do I stop this thing? Go back and here. No. Okay, back to the web. Okay, second example, aptamers to block and monitor, and block virus contamination. So it's quite of the thing of the day. So this is ideas for a project to go on from the first project on EPCAM. on electrochemical nanosensing, but applied to the spike protein of the SARS-CoV-2 virus. So this is an email that I sent to Nicolas and Chan on March 27, 2020. So you see the structure. We have a gold surface, the linker then there is the ferrocene, that is again the current detector, the current producer, let's say, and then another linker to the aptamer that has to go to interact with the spike protein. Okay, so look at the colors of this figure, and this is a preprint that appeared in October 2020. Even the colors are identical. Of course, they didn't copy us because the email was secret, but I can see that I had quite a future foreseeing in this thing, because the idea is exactly identical to what we were kind of proposing in this initial project. So how do we go about this? Well, in this case, there are no experiments done, so it's completely theoretical. This is the well-known structure of the spike protein. By now, I think that almost everybody knows what a spike protein is. And the key element of the spike protein is this cyan domain here, the RBD, receptor binding domain, which can be in two different conformations, closed and open. When it's open, it interacts with this ACE2, which is a cell receptor, and then the protein binds to the cell receptor, and a process starts down here in the S2 part of the protein that is opened and cleaved, and this opens the way for the viral RNA to enter and zip into the human cells. Okay, so this is the key event, basically, right? The opening and closing of this domain that leads to the first interaction with the human cell receptor, and then to the proteolytic opening of this part of the protein. The two configurations are quite freely fluctuating, so there is almost no barrier, no free energy barrier, and X-ray and cryomicroscopy data show that these two configurations, open and closed, quite freely fluctuate with about 50-50 occupation probability. So right after the Wuhan 2019 events, people started working on this, and a Chinese group identified in Hong Kong, identified two different aptamers that they called A51 and A67 that interact with the spike protein. So they proved the interaction, they demonstrated the interaction, measured a very small, very low, not small, high and very small sensitivity in the nanomolar range against the RBD. So the experiment was performed by cutting away only the small RBD domain, this cyan part, and they used this domain to fabricate, to produce these two aptamers. So then we come on board, and we try to redo, to assess how this binding works. So we do our procedure. We first do the docking of the red aptamer here on the complete protein. Here you don't see the rest, but this is only the RBD and the N-terminal of the protein that is used for the docking. And then we do the full molecular dynamics with the whole protein here at finite temperature that reveals how the aptamer interacts. And one important thing, I will show it in the next slide, is that this interaction is also blocking the opening. And then we can go on and do measures of free energy and characterize the hydrogen bonding and so on and so forth. So we can have a ideally perfect knowledge of what's going on with this aptamer interacting with the spike protein. One key event that we showed, that we think we showed, that is very important, is that the aptamers interferes with the ACE2 binding. Although it doesn't cover completely the RBD domain of the protein, it covers enough to block the interaction with ACE2 and probably to slow down or even block completely the opening of the receptor binding domain. So in that case, the the binding of ACE2 will be completely suppressed. So here on the right, on the left, you see the experimental configuration when the spike interacts with ACE. You have these balls here that indicate the interaction surface. So you see it's a lot of interaction here. When the aptamer is there, it protects, let's say, the RBD. So the ACE2 approaches, but it cannot quite freely interact, as in this case on the left. And the result is a very modest interaction here on the right, and interaction with the DNA. So the DNA is directly chasing away, let's say, the ACE2 from the interaction, much reduced interaction with the receptor binding domain. So this DNA aptamer can be a quite effective solution, both for recognizing the spike protein and for therapy, if you want, something that could stop or slow down at least the development of the virus penetration in the cell. On the way, we learned a number of interesting things. For example, DNA interacting with glycans. Now, the spike protein is a heavily glycosylated protein, and nobody knows how DNA interacts with sugars. Why? Because in nature, DNA practically never has the chance to see a sugar inside the cell nucleus. RNA, yes, because RNA goes outside the nucleus, but DNA stays all time inside the nucleus. So that's a prime, if you want. It's uncharted territory. And so nobody really knows what's going on here. We observe a number of hydrogen bonds that form at specific regions between sugars, mannose in this case, and DNA. Here we have a repetitive structure that binds the OH hydroxyl oxygen of mannose to the backbone of DNA. You see, it always binds to the O2P oxygen of the phosphate. So maybe it's just a chance, because I must tell you, there are no database of interactions, of force field interactions that today are optimized for this kind of calculation. So we are guessing. But maybe it's a lucky guess, because more or less interaction could be, I would say, 60–70% realistic. And we are here in the process of discovering a new territory, entirely new territory, in describing the interaction of DNA with sugars. So that's a really new thing. So let me quickly conclude with this last part of the story, aptamers as proxies for the RNA world. We are not working on this yet, so I'm just giving you some ideas, but I think it's fun. It's really interesting part of the story. So we are in the Archean Earth, 4 billion years ago. One prerequisite for the emergence of life, whatever road the life would take or has taken, is that at some moment, some information-carrying polymers must emerge by purely chemical means. This is one key step. Now, in 1966, Sol Spiegelman, that we already saw in the photo in the beginning, reported that RNA could synthesize himself. That was the first step in a long story that is really amazing, but RNA can serve as template for synthesizing new RNA identical to itself. So it's capable of auto-synthesis. A few years ago, Temin and Baltimore discovered an enzyme, the reverse transcriptase, that can make DNA from RNA. So normally in cells, we have the transcriptase that make RNA from the DNA to then make proteins and so on and so forth, okay? But this was a key discovery for the RNA world theory, because it proves that DNA could have come after RNA, and then the first RNA is self-copying itself, and then can start the process of making DNA via this kind of enzymes. So we need just a few more tools to start experimenting with in vitro evolution. And this is what the people that I showed you in the first slides were doing, trying to invent some technique that could generate millions of sequences at random and automatically select the most interesting one, where interesting in this case means sequences that are capable of auto-producing and auto-catalysis. So to make the building blocks of the, and then Selex came out between the 80s and 90s, and this was the key step for having an ingenious way to amplify, mutate, and select RNA molecules, trying to accelerate in the lab what should have been happening over 500 million years in the Hadean Earth. So we are ready to start playing the game of evolution, okay? So Jack Szostak, Nobel Prize for Medicine in 2009, again, started a number of experiments where he tried to identify which RNA could catalyze reactions for self-replication. So you start from small sequences, but then you won't end up with a genome. So one key step is to ligate, to join together these molecules and then copy them and do all the rest of the business, okay? Now, the number of possible reactions, of course, is huge, intimidating. And the number of conditions that can vary, pH, ions, temperature, pressure, is even more intimidating, okay? In all cases, first we need activated nucleotides that start making short chains. This could have been possible in the old Earth on some kind of organic or maybe inorganic templates, these under sea vents. That's one of the possibilities. And then we need to start building here, you see, membrane. This cannot come from acid, but membranes can assemble quite spontaneously from lipids, and then packaging this primordial RNA into a kind of a lipid protovesicle could be the first step to a kind of cell if the conditions are right, and that's the big if. So Szostak started a very key experiment in 1993, which today is replicated in many laboratories around the world, and he was able to reproduce both conditions. And the key thing is that in his experiment, he started with the SELEX method, actually, and the goal was to see if starting from a completely random set of molecules, one could identify some subset of molecules with specific properties. And these properties was the ligation that you see here, the fact that you start with small sequences and the sequences can ligate together in longer sequences. Now, in our life, in our cells, we have enzymes that do this. But in the old Earth, supposedly there were no enzymes. So RNA is shown to be able to both self-copy and do self-catalysis. So the RNA autocatalytic activity, just after 10 rounds of amplification by SELEX, increases by 8 orders of magnitude. This was the key experiment that showed that this whole procedure makes sense. And it's from here that the RNA world idea started, okay? So what this experiment tells is that once you have a small population of catalytic molecules in a huge population of random mass, then this will start and inevitably grow and take over all the rest. And these self-catalytic molecules will copy themselves. So in a sense, they are transmitting information to their progeny, okay? And then let's go Mars. RNA world on Mars. Now, what's the big problem here is that RNA is very unstable. That OH group, the hydroxyl group that I showed you before, is easily catalyzer for cleaving the bonds. So it would just disrupt the RNA just formed. Which factors can stabilize RNA long enough to transmit and sustain life? I should put quotes on that life word. So the experiments suggest at least the following, that RNA is chemically stable between pH 4 and 5 and at close to 0 temperature, 0 degrees Celsius. And divalent metals, as we already saw, magnesium, manganese, iron, facilitate folding into stable 3D structures. So the problem is that on Earth, we don't know what were the prebiotic conditions about metals, about pH, and so on, because the geologic events on Earth have cancelled everything. It has been wiping out all the rests of the ancient world. But we can search for environment consistent with this RNA stability on Mars. Why? Because Mars is a planet that is frozen in time and is more or less preserving the primordial surfaces which have remained practically unchanged since their formation. So maybe there one can find an Mg2 atmosphere coming from the basaltic surfaces, weather to slightly acidic waters around pH 5, and maybe this could be the stability optimum for RNA on Mars, and maybe this could give us hints on what really could have been happening on the ancient Archean Earths. This was proposed in a very recent paper, again by Jack Szostak, which maybe wants to win the second Nobel Prize this time. He proposed a series of experiments to do on Mars to try to look and find for this kind of conditions. So conclusions, I think I showed you that aptamers may represent a great potential for label-free screening of several things, cells, viral particles, and cancer cells. Detailed knowledge, this is the key point where I would like to make a contribution as a theorist, as a modeler, simulator. Detailed knowledge of the microscopic interaction is key to understand better the more precise definition of what the aptamers are really doing. So, for example, we showed that in the EpCAM cancer cells, now we understand much better the Brownian dynamics of the current carrier and how the aptamer interacts with the surface cell proteins. So I think we make quite a step forward with respect to the blind experiment of directly measuring the current without knowing what's going on. And for the SARS spike aptamer, we discovered, let's say, that the interaction of the aptamers can have also a therapeutic application in principle, because it blocks the opening of the protein and keeps away the interaction with the human cell receptor. This domain of engineered self-replicating nucleic acids sheds light over key steps in the origins of life, and maybe there is work to do on computer modeling also in this case to try to understand the evolution and the growth of RNA as an auto-catalyzing molecule. So acknowledgement, of course, we use lots and lots of computer time, of supercomputer time, that generously is offered by the two National Supercomputing Center. Some projects give us some money, not very much, but we are theorists, so we don't need very much, just to pay 1 or 2 students every now and then. And thanks to you for your attention.
Thank you very much, Fabrizio. Very interesting and new, at least for me. So please feel free to ask questions. That's why we are here. So there are no stupid questions. No. Just go ahead and feel free to ask.
Can I ask a question?
Yeah.
Thank you very much, Fabrizio, for this great talk and your amazing work as a biologist. It's always amazing to see how many interactions you can do with all the molecule, ions, protein, RNA, etc. So it's really amazing for me. I do have a question. We know as a biologist, we know that protein can interact and have an effect. We know that RNA and DNA can interact. We know that protein and RNA can interact and have a function. We call that RNA binding protein. And most of the time it's inhibiting the translation of the RNA. We know that RNA and RNA can interact and inhibit also. We call that miR in nature, miRNA, microRNA. And we use it to do siRNA. And here you are speaking of aptamer, which can bind protein and affect the function of the protein. Do you know any example in nature where RNA can modify the activity of a protein?
No, I don't know any examples. What I can tell is that the peculiarity here is that aptamers are made from single-strand DNA. And this rarely occurs in vivo, I think. Am I right? And that's the key point. When you make single-strand DNA, it folds onto itself and it becomes a sort of RNA kind of molecule with little differences because it's still a DNA basis. But from the 3D structural point of view, it really looks like an RNA. So it can perform many of the functions that RNA plays in the cell. It can bind, it can do more or less whatever RNA does, with some differences, of course, because it's not exactly RNA. And the advantage is that it's much more stable than RNA, so it lasts longer. So I would say that a simple answer, I don't know examples in nature, but a simple answer is that single-strand 3D folded DNA can do pretty much, let's say, 80% of what normal RNA could do. Normal small RNAs could do, more or less. From a structural point of view. And functional, it depends on what protein, what's... Okay. But that's the big similarity.
Okay, thank you so much.
Thank you. Any other question? Christophe, peut-être? No.
Yes, I have maybe a stupid question.
No.
You mentioned aptamers for therapy. Are the aptamers already used for therapy for some diseases?
I don't think there are. I think there are 2 or 3 cases in which aptamers have been approved by FDA in America. There are a large number today, more than 200, I think, aptamers that are characterized against specific proteins. So they could be used. They are ready, let's say, to be used. I don't think that many of them, I think maybe 2 or 3 cases, they have gone through the whole approval procedure for therapeutics. There was a case for, I think one was for avian influenza and another one for something. But I think it's only 2 or 3 cases in which RNA aptamers have been approved for as drugs. All the other ones are being actively developed. There is a whole community that works on this thing, but still, I don't know if, I'm not an expert, I don't know if there are some particular procedures linked to the fact that being nucleic acids injected in the human body, somebody may think that they could alter the genetic material. It's quite unlikely because these things don't go in the nucleus. They have no way of going to the nucleus unless something is transporting them into the nucleus. But nevertheless, you have pieces of RNA or DNA circulating in your body. Now we have, because I've done the Pfizer vaccine, and you too, I think. So now we have pieces of foreign RNA circulating in our bodies. So maybe this is just the first step for aptamers to win the race. It's kind of scary, huh? No, it's not.
Don't say that.
I have a question, Fabrizio. When you said that in your structure, in your computer program, you say that to predict the 3D structures, you have to convert DNA to RNA. I am not at all in the field. Why do you have to do this? Why?
Because there are predicting the 3D folding is today still a nearly impossible task. There was of anything, of proteins, of DNA, RNA, and so on. So RNA is easy because it's just a double helix. So we know everything about DNA, but that's not the thing where if I give you a sequence of amino acids, nobody can predict what is the final structure of the protein. And this is true also for DNA and RNA in single sequence. There is one code that does, with some good degree of success, 3D folding of RNA, and that's it. That's the only thing that is really available on the market. And so what we do, we start from our DNA, we do the 2D folding first, then transform this 2D folding in RNA folding. It requires to replace the base T with U and adding the OH at all the sugars. And then we pass this structure in this single unique code that is able to do a 3D folding of something, of RNA in this case. Then we get a putative structure and then go back to DNA. So replace again the bases and remove the hydroxyl from the sugar. Of course, it's not a 100% safe procedure because but in the end, nobody really knows what's the real fold. So the only possibility is to do this procedure and see if it works, as I showed in some examples. If you're reasonably sure that it works, then you extrapolate and say, maybe it will work also in the N+1 case that we are going to do. So that was the, I have to quickly go back to this slide. This one, where you test this procedure on something for which there are X-ray data available. Kind of work, of course, is not perfect, but we don't know if it's not perfect because the initial 2D structure was not exactly correct. We don't know if it's not perfect because the experimental data are not correct, because these are very small chunks of RNA, so you don't know if the experiment is so capable of getting to this refinement. And the other idea is that the passage between 2D and 3D is quite tricky because the 2D is kind of blocking us the hydrogen bonding interaction. So in 2D, you decide that C interacts with G, A interacts with T, so Watson-Crick, and then you make the pairs. But this is based only on the nearest neighbor model. There is no folding energy in it. When you go to the 3D, you have the whole mechanical structure. So you have elasticity, you have electrostatic interactions at long range and everything. And you are not really sure that what you input as best 2D will stay as a best 3D, because there is no possibility of interplay between the two. So it's all based on some on-the-fly assessment, hoping that the final result is reasonably close to the reality. Of course, the final check is this. If we could have for each aptamer an X-ray structure, then it would be, but it costs lots of money. And as you see, people produce aptamers in, tons of variants. So one cannot possibly do X-rays for any variant because it would be just impossible. So maybe it would be a step necessary for the FDA approval, for the safety approval. At that point, if you guys, you have something that works, then show us the X-ray and let's see what it looks like at least.
Mm-hmm. So what was the challenge? I mean, you have to develop new, to improve these simulation tools, this predictive tool is what? More calculation power, or use neural machine learning, or what is it?
Maybe, yes, maybe machine learning or this kind of a technology. Okay. Yes, maybe one of these techniques, yes, because the one thing that I think it's a little bit broken in this process is this having to break the process into different steps. If we could have one single tool, whatever it is, that is able to start from the sequence and jump directly to the final 3D structure in one shot, that would be the big step. This is true also for protein folding and not just for aptamers folding. People have to break the process in many steps, and from one step to the next, you introduce a number of hypotheses, a number of approximations, a number of cutting corners, and that makes the thing less and less and less reliable at each step. At each step, you're introducing noise in some way. So the final result is affected by all this intermediate noise. maybe, yeah, machine learning or some techniques like that, artificial intelligence. There was a claim a couple of months ago, I think, that an artificial intelligence machine has solved the problem of protein folding. Huh? It was in the it made a big noise in the literature and in the press. I don't know if it's true or not. We'll see. But maybe this is the way to go. Yes.
Okay, thank you. Other questions?
Yes, if I can, yes.
Yeah, sure.
Yeah. Okay. First of all, thank you very much, Fabrizio, for this very interesting seminar. And my question is, can you discuss a little bit more in details the quantitative measure of the affinity between the aptamers and the other biological objects?
Experiment again. Experimentally or theoretically or both?
Well, I think theoretically, but both is more interesting. If you can.
Yeah. So experimentally, they just go on more and more diluted solutions until they still see interactions. Basically, the idea is this. So you do chromatography, titration chromatography, and at some moment you see nothing more. And then you see that that's the limit, okay, of detection. Computationally, the basic information is, hold on a second, it's here. So it's in the free energy calculation part.
Okay.
So what one can estimate is free energy barriers for attachment and detachment of the thing. And from the integral of these free energy barriers, you get an idea of how strong is the bonding. Then the exponential of minus delta G over KT gives you the KD of the solution. For example, in our case, we found numbers that roughly are in the same order of magnitude of the experimental values. In the 10, we find 10 to 30 nanomolar, while experiment is more in 9 to 20. So, but we are in the same ballpark, let's say. So that's the basic idea. Of course, this is affected by which free energy method you choose, what kind of free energy calculation. But what we do, it's more easily umbrella sampling, and then weighted histogram recalculation to extrapolate the free energy barriers. Of course, it might not be the best idea, because all these free energy methods, as you know, even better than me, they are affected by the reaction coordinates that you choose. So metadynamics is a little bit better, but in these complex objects, also metadynamics, you don't know where to go, basically. We should need a method that really is more free, that doesn't force you to choose particular reaction paths. Okay.
Okay. Thank you. I have another curiosity about the origin of life of the biological molecules. How we can reconcile the experiment of Oparin and Aldan, which were more on the amino acids and proteins, with the procedure, the research development you have presented, which is based more on the nucleic acids. I want to say it's we have to consider before the proteins or before the nucleic acids in the origin of macromolecules.
Well, that in the 20s, that was quite an old debate, because it was not sure who was coming first, who was carrying the key information. Okay, of course, amino acids have to be there at some moment in the evolution of life. Who comes first to start the whole procedure at that time was not yet clear in the 20s. It became clear in 1952 with a key experiment that showed without doubts that DNA and RNA are the carrier of information. This is before Watson and Crick. And from that moment on, we know that RNA, DNA must come a few days before amino acids. So amino acids must arrive at some moment, and they would develop maybe through the Oparin experiment way, or that other, I don't remember the name, experiment that was key in the
Aldan, maybe.
No, there was also another one very interesting, IEMN Miller, okay. The experiment with her. But that would be parallel or after, I think. The first step is something capable of self-replicating. That's the current view. Then what is this self-replicating thing? There are people that are thinking of mixes of peptides and RNA. Because it is not so 100% sure that RNA catalytic activity can be strong enough to support real ligation down to the end of the road. There is a very interesting paper. I will send it to you because you will surely like it. There is a very interesting paper by John English at Oxford, where he started a new idea about this self-catalysis thing based on the fluctuation dissipation theorem. It's very interesting work. Very, very recent. It's like 2 years ago. He's still working on that.
Thank you very much.
Thank you.
Thank you, Fabrizio. Any other question? Okay, so if not, I think that we can stop here. Thank you again, Fabrizio, for giving this very nice talk. This talk is recorded, so I will put it online. Well, not online. We'll send you a link through Renater. Well, maybe this link can be put on the IEMN website. So have a good day and see you on the 23rd of June with the next internal seminar from Zim Optic. Thank you very much.
Okay, thanks to you.
Thank you very much. Great. Have a great day.
Thank you.